what I'll do a bit more of today. Okay, so I'm going to tell you about a few things that I've worked on to try to put them in a general context, not just all about me. And so what I want to focus on in particular is how does selection work in spatially structured populations. Now, we touched on that a little bit last time when we, well, a lot last time when we talked about the conditions for maintaining variation via spatially heterogeneous selection. Right? So that's obviously probably the most important part of how selection and spatially heterogeneous and spatially structured populations interact. Is it creates this path for divergence among populations. And one of the things I didn't emphasize last time enough, and I'm not going to talk about much today, but I just want to, in a sense of good pedagogy, emphasize, at least briefly here, is that that pattern of genetic variation created by effective local adaptation is probably one of the main sources of maintenance of useful genetic variation in populations. Right? So now that's we still don't know what fraction of genetic variation is maintained by input of mutation per se, or what fraction is maintained by spatially heterogeneous selection, or other kinds of ways to maintain variation. But it's clear to me that spatially heterogeneous selection is very important, at least in many or most species. And I'm not going to talk more about that. I just wanted to make sure that I conveyed that feeling to you, because I think it's very important. And I want to add one other comment to that, which is to say that unlike the kind of variation maintained by some other mechanism, especially the variation maintained by mutation selection balance, the alleles that are maintained typically by spatially heterogeneous selection are more useful, are likely much more useful to the population under future circumstances than the alleles maintained by mutation selection balance. Mutation selection balance are bringing in bad alleles. Those, of course, may be beneficial under some future environmental context, that even if they're deleterious now, and that's clearly the case, that will happen. But spatially, alleles that are maintained by spatially heterogeneous selection have adapted to some aspect of the environment that exists. They've been tested, and their effects that benefit the organism in that environment are greater than the deleterious pleiotropic effects of those alleles on fitness in other ways. So they're a good set of alleles that can matter quite a bit, right? So even if they don't I believe they make up a lot of the genetic variance for fitness, but even if they don't, they're more important in terms of future adaptation than you'd expect just by their proportion of the heritability. Okay, so just wanted to say that. I don't have anything else to show you with that, but I think that's an important thing to think about. And I think actually there's a lot of potential for research directions associated with that. Okay, so I'm going to come back later to talk about local adaptation later in the talk, if I get that far, in the sense of thinking a little bit about how do we measure, how do we look at the genetics of local adaptation from looking at genomic data or quantitative genetic data. But first I want to start with thinking about selection, not in the context of this spatial heterogeneity, but just as a baseline, how does selection, even uniform selection, affected by the existence of spatial population structures? So that'll be mainly where I start from. So I'm just referring to that here as simple selection. It's a homogeneous selection on a single locus with simple genetics. How is that affected? How is the selection on that locus affected by spatial structure? And there's a lot of, I think, cool things that happen there. And then I want to spend a chunk of the talk toward the end about how do we identify traits and genes that have allowed local adaptation. And maybe I'll come back to this theme that I talked about yesterday, about how do we interact with the assumptions of the methods. So that's kind of in a way a sub-theme of all of this. Right. So let's talk about uniform selection and metapopulations. So here's the goal. We want to be able to model selection in spatially structured populations. We want to use measurable quantities, if possible. So something like FST is a measurable quantity. And we want to allow for, we want to get away from the island model. We want to be able to think about what happens, if possible, in the absence of assumptions related to the island model. And in particular, it'd be nice if we were able to do that without having to know all the details. And I'll show you a little bit about how that might work. So I'm going to show you that we can get pretty far in understanding selection in spatially structured populations with three quantities. One of those is readily measurable. One of those is in principle measurable, but it would be fiendish to actually do. And the third one is something we just don't know enough about in spite of 75 years of population ecology, although we ought to. And they are these. So how does genetic variance partition across populations? We'll measure that with FST that we talked about yesterday. What's the effective population size of the metapopulation? Okay. So today when you see NE, I'm going to be using that NE to refer to the effective size of the whole collection of populations, of the species that we're looking at. And that's, so FST is the one we can measure readily. NE is the one we can measure in principle, but it's challenging. And the one that we really need to know more about but don't is what's the relationship between the local fitness on a genetic scale and the productivity of a local population. What's the relationship between the genotypes of the individuals in a population and how much that population as a whole contributes to the next generation? Okay. I'll explain more about that as we go. And that'll come up with a parameter B later on that I'll explain when we get there. That's one you'd think we'd know a lot about, but when you talk to the ecologists, they go, oh yeah, that's a good question. I really wish we knew that. But I'll talk a little bit more about some of the things we might infer about this B. Okay. Right. So there's a couple. I took a slide out, and the also is lost. So FST, remember from yesterday, does two things. It tells us about the variance among populations. It tells us about genetic differentiation, and it also measures the decrease in homozygosity caused by that population structure. And both of those things turn out to be important when we're thinking about how spatial structure affects evolution. Okay. Now this B, this parameter to ask about the relationship between genetic fitness and the absolute fitness of a population. This again, something we don't know much about. There's a lot of possible relationships between the genetic fitness and the contribution of a gene. Yeah, I will, just a second. Yeah. Well, I'll go right now, sure. So what I mean by productivity is, in particular, when we get down to the equations of this, when you count up how many individuals in the next generation came from parents that were in this population. Okay, that's the product. That's what I mean by the productivity of the population. So the fitness, as we usually define it in, how's that different from the fitness, he asked, in the parental generation? The fitness, as we usually define it in these population genetic equations, is a relative fitness, right? So it's saying how good do you do relative to the other individuals in your population. Well, that's not speaking to the case of, well, how well does that population as a whole do? Okay? So it's possible, and let me add this jargon in, and I'll try to continue answering that. It's possible that the population has a productivity that's predictable by the genetic quality of the individuals that are there. In other words, that the productivity of the population, the contribution of that population to the next generation, may be proportional to the relative fitness of those individuals relative to the mean fitness of everybody in the whole species. Okay? That's what you would expect it to be if there were no actual local interactions between individuals. If you could just determine how many offspring a genotype with big A was, regardless of where it was, how many actual successful offspring it would have, and you didn't have to know the local spatial context of that individual. That would be this first case. That is we call hard selection. These terms, hard and soft selection, are probably not extremely, well, they're horrible terms. But they're used enough, it's probably worth knowing about them. They come back to Bruce Wallace in 1968, Brian mentioned the other day. Hard selection was when the actual genotype of the individual affected the success of the population. Okay. Soft selection, in contrast, is another point on a continuum, but it's an interesting one to talk about. So soft selection is where the productivity of a population is independent of the genetic quality of that population. Yeah. It's really tough, right? So if the difference between a big A and a big A small a was the same everywhere in terms of the actual count of individuals, then that would be hard selection. If a population of size n always contributed a certain number, regardless of what the gene frequencies were there, that would be soft selection. Okay, so you can imagine. Let me give you a concrete example of this. For soft, a classic example of soft selection, whether it's true or not, but it's a nice cartoon that describes what it is imagine that you have, maybe I said this the other day, you have tree holes that are nesting sites for some bird. And those they can't create those tree holes. There's a finite number of those, and they have to compete for access to those. So a population that has 100 offspring in a place where there's only 20 tree holes, only 20 or 40 or whatever, however many go together in a tree hole, can succeed no matter what, right? So you can have the best genes in the world, but if you're limited by the number of tree holes for the productivity of the population, that's what it is. You're competing locally for access to those tree holes, and that constrains the productivity of that population. In contrast, you might some other resource may be more fungible. You may be able to become more efficient at using a particular food item, and so a population that had a higher fitness may create more offspring even over the same number of resources, and that might be more like hard selection. The better quality genotypes would create more offspring even if they were with limited resources. Okay. Yeah. Absolutely. Yeah. Now these are not terms that apply to a population. So you're saying they don't stay constant over time, they could change? It's worse than that. It's not the same genes, different genes, different loci in the same population may experience more hard-like selection, and other genes may experience more soft-like selection. So we were talking the other day during the mutation load lecture about the importance of when the gene was expressed, when it was acted on by selection. Something that acts early is going to act more like hard selection. Something that acts late, after population regulation, after your resources have been used, is going to act more like soft selection, perhaps. Right? This is again a really open area of research. The kind of interactions between the kind of mechanical ecology, the specifics of the way genes act, and the way they constrain the relationship of who competes with whom is something that we still need to work a lot more about. We've done some stuff about that. But so this is not a thing that, it would be nice if this was a thing where we could say, okay, this species, nuthatch, soft selection. This species, wheat, hard selection. It's not like that. It's really going to be down to gene by gene. So like I said, it's hard to measure. We wish we could measure it. It's potentially measurable, but it's a hard quantity. So So I'm going to use this B to scale between these hard and soft selections. Okay? These are 2 points on a continuum. They're kind of instructive points, but they're not biological in any sense that they have to be one or the other. There's a continuum, and there's also things kind of off this continuum that are more complicated than I have time to talk about right now. But these are interesting points that are instructive to think about. More questions? We good for what these things mean? Okay. Yeah, well, so here's a little cartoon of that. So with soft selection, so here these clusters of dots are different populations. The red guys are better than the white guys. The red guys have 50% more offspring. But with soft selection, you have local regulation. These are competing for access for local resources. So they each only make 6 offspring, in spite of the fact that these guys made a lot more, had the potential to make a lot more. If they were all mixed together, the red guys would have increased in frequency over that period of time. In contrast, with hard selection, these guys increase in frequency relative to those, just like above. But with hard selection, they're allowed to contribute to the next generation in proportion to that increase in fitness. Okay? Now, that can be because this population grows. It could be because this population contributes more migrants to other populations, right? So don't think about populations necessarily being static in their contributions or in their size over time. You can allow hard selection if individuals just ignore who they're around in terms of what their contribution to the next generation is. Okay. This notion of hard selection I found can be very difficult for people who are stuck in the population genetics theory to get, because they know population size is n. How can they? No, it's not n. N's the thing that changes, right? And it might change because of fitness. Okay. So sorry about the names, hard and soft selection. They're horrible. In 3 minutes you won't remember which one's which, but don't feel bad about that because that's true of every single person in the world. If you remember in 4 minutes, I'll give you a prize. Okay, so here's just a summary of some of the stuff that we've done about this. With these parameters, sometimes with just a subset of them, you can predict the changes that population structure causes to kind of core population genetic quantities of things like mutation load, inbreeding depression, probability of fixation, new alleles, mutation drift balance, blah, blah, blah. I'll tell you about a few of these things that will try to show you some of the reasons for the changes and the magnitudes that might be. Okay, so I'm going to, when I said in the title, simple selection, I kind of meant it. This is almost as simple a selection model as you could have. Lindy's not here, so I can't play the haploid. It's not haploid, it's diploid. And we've got the potential for a dominance coefficient for the heterozygote. So those are our token efforts toward complicating the selection function. We're going to assume for all the calculations in the first half that selection's weak, and I mean that in 2 senses. We're going to assume that selection's weak relative to the migration rate. All right. Now that's a severe constraint on the application of this. Sometimes selection is quite strong, but this will be true for most loci. So selection is weaker than migration rate. The reason for that assumption is because when selection is weaker than migration, we can actually predict the patterning of genetic variation over the landscape from neutral models and get a very good idea of what, for example, FST is, even for a locus that's under some selection. Okay. If selection is stronger than migration, that's not true and becomes a much harder problem. Okay. So that's going to be one of our useful approximations, is that with weak selection, the neutral FST, now the average allele frequency is going to be strongly affected by selection, but the patterning, the relative standardized patterning that's reflected in FST is not changed much. The other sense in which we're going to assume selection is weak is that there's a Taylor series coming up in S, and so it's also going to be small relative to 1. Okay. Now, okay, so let's just, at broad terms, think about how will response to selection be affected by spatial structure, even with a simple model, right? So we've already assumed away one of the ways that selection is affected by spatial structure is that you might have different selection pressures in different places, right? We're not doing that. We're just saying uniform selection. But even then, even with uniform selection, spatial structure does two things to affect the response to selection. So one thing it does is that it can reduce the efficacy of selection because in the event where you have some version of soft selection. Well, let me explain that. I don't think I did before. So if you have soft selection, right, you're competing with other individuals nearby for access to the next generation. Okay, now, a key feature of this is those other individuals are genetically similar to you. Okay? So if you do well, you have offspring. Those offspring are going to be consuming resources. They are going to be taking a share of the next generation. And they are reducing the potential for other members of your population from having as many offspring, and vice versa. They're affecting you and reducing your probability of having offspring. Those other individuals are related to you more than you expect by chance because we have population structure. So when you do well, you have a negative effect on other individuals who are like you, who have a similar genotype. Okay? So what that does is reduces the effective response to selection, effectively because of competition between relatives. Okay, so if you have population structure, and when I'm using structure now, I guess in a more narrow sense of you have structure genetically of the population, so that there is a positive FST. That will effectively reduce the response to selection if you have something like soft selection. Yeah, I would take out the only if, but it's horribly confounded when the selection is interacting with the type of regulation, right? So so David Van Dyken, who's a postdoc with both Michael and I, Michael's got him more than I do because he's there now, but has done a lot of work about following the particular patterning of how this competition works mechanistically and how that makes the dynamics of these things change. Yeah. So if you grow food and then everybody has access to that food, then that's temporarily relaxing the competition between relatives, and it behaves differently from what I'm talking about. Yeah. Okay. Those are cool papers. MNAT. Okay. Okay. So that's it. There's one way, right? So population structure can reduce the efficacy of selection. But on the other hand, as we mentioned yesterday, population structure increases the frequency of homozygotes. And if you have more homozygotes for the same allele frequency, you're going to have more effective selection. And kind of for two reasons. These kind of overlap into maybe a fractal number of reasons, like 1.7 reasons. But, it's this. So if you have more homozygotes for the same allele frequency, you're going to have more individuals which are more different from each other, right? So big A, small a population like that has more genetic variance, even for the same allele frequency, than one at Hardy-Weinberg with all these intermediates. Okay? So selection can act on that. It's more effective at removing the bad alleles or increasing the good alleles. Okay? And related to that, a subset of that, but of special interest, we've talked about it, it's a very important concept, is that evolution causes the expression of homozygotes. And if the allele is rare, if there's a rare recessive allele, then selection can actually see that more effectively, like we've mentioned before. And then, again, selection is even more effective in that case. Right? So even with additive alleles, even with HEs with half, you'll get more effective response to selection for this reason, but just because there's a bigger genetic difference between the two homozygotes than there is between the three genotypes. But on top of that, there's an extra special effect when you're expressing rare recessives as homozygotes. You get a much more effective selection. Okay, so that's the second case. All right, so we have one thing that decreases the efficacy of selection. We have one thing that increases the efficacy of selection. So it's not obvious which one of those would win, which one's more important. Let's do some theory. Okay. So if you do that, there's steps before this, but I'll just show you one of the intermediate results. And I'm not really going to actually work through this too much. These are the two special cases for hard selection and soft selection. What I'm showing you here is the allele frequency change of the average allele frequency over the whole species caused by selection. Okay. So that's a function, and this is an approximation for weak S, where the Taylor series happens somewhere in the invisible background. So we have the allele frequency, the average allele frequency here, selection coefficient, dominance coefficient, and we have FST. Okay. So that has all the same things you'd expect in a pan-mictic, randomly well-mixed population, but it also has FST, and it gets more complicated when it does. Right? The other thing it has, and this is the fun part about it, it doesn't have all the little QIs and PIs from all the different populations. You can express it in terms of the average allele frequency and how that will change over time. And the kind of nice leap forward here is that you can describe the effects of that heterogeneous PI and QI with FST in the equation. Basically comes from looking at the moments of the allele frequency change, and then if you mix that around, you wind up with a variance in allele frequency that you can then express as P bar Q bar FST and resolve to this. Okay, so that FST is not going to be changing much as a function of Q bar. Okay? It's a quantity that's relatively constant. We can understand the dynamics of this in a much easier way. Similarly, for soft selection, you can do the same thing. It's the same equation as with hard selection, but it's got this extra term in front, 1 minus FST over 1 plus FST. Okay. So here's the effect of that competition between relatives, right? So in the sorry, I can't see if I just block it, but here's the effect. Here's what happens just because there's population structure with no local competition, right? Hard selection. All that's going on, right? The differential expression of heterozygotes. All that's going on, but on top of that, with soft selection, you've got a constrained amount of response to selection that's caused by competition between relatives. And that's decremented by this 1 minus FST over 1 plus FST. And for those of you who've ever seen the social evolution literature where they talked about relatedness, that turns out to be 1 minus the relatedness of local individuals, that whole first term. Okay. Questions? Are you guys hot, or is it just me because I'm flapping my arms around? It's just, okay. Don't have to. If it's only me, I'll live. Okay, so I'm going to show you a few slides that show some of the effects of this using these calculations. I'm going to just show you to them graphically. And there's 2 or 3, 4 slides that have the same kind of format. Thank you, Deepa. They have the same format. So I'm going to walk you through the framework here before we talk about the biology yet. So this axis is FST, starts at 0 and goes up to a ridiculously high number. This axis is going to show a relative quantity. Okay, it's going to show a quantity, whatever we're interested in at the time. So in this case, I'm going to be looking at the equilibrium mutation selection balance allele frequency, Q hat. But relative to what that would be in a population that was pan-mictic, that was completely well mixed. Okay? So then on every graph like this, these should converge at 1, or else I did my job wrong, because at FST equals 0, that's that case where everything's undivided and well mixed. Okay. Then I'm also going to show you the results for 3 different dominance coefficients. And I'm going to show you results for, the labels are kind of reversed, for dashed lines are hard selection and soft selection with the solid lines. Okay, so there's a lot there. What we got? We got soft, hard, H equals 1/4, H equals 0.1, H equals 0.001. 0.01, sorry. So here's something close to additive, and down at the bottom, here's something with a fairly low dominance coefficient. Okay? Right, so let's go back to the biology of this particular slide. The equilibrium allele frequency is always reduced when you have hard selection. Okay? So when you have hard selection, you don't have this counterbalancing force of competition between relatives that hinders selection. But you do have this more effective expression of homozygotes. The effect of that effective expression of homozygotes is not very great in the case of this nearly codominant locus, but it's quite extreme when the locus is nearly recessive. Okay, so here this dramatic effect by expressing the alleles as homozygotes more often. As you get even a little bit of FST, you get a dramatic reduction in the equilibrium allele frequency of these deleterious alleles. Okay. Good questions? Okay, well, let's translate that into mutation load. So here's mutation load. So this is a load calculated for the population with its mating structure. So with its value of FST, what would you actually see as the reduction in mean fitness relative to an unloaded case? That's going to be a little bit higher for the populations that have structure because they're going to be more often expressing these deleterious alleles as homozygotes. And again, you see that with hard selection, you get strong reductions in load. With soft selection, you get reductions in load for small values of FST, but then for most values of H, that load increases with population structure because the competition between relatives term takes over, on top of which the fact that you're expressing these things in a homozygous way creates a lower a reduction in fitness. For the same allele frequency, you're reducing mean fitness. I should point out that this graph is one of the horrible graphs. You shouldn't do this. I shouldn't do this, but I did it. That doesn't stop at 0. It stops at a half at the bottom. So it looks more dramatic than it is. It drops by a half at most. Okay? And that's consistent with what you would have predicted from what we talked about on Tuesday, that because when you are under these situations down here with the low dominance coefficients, alleles are being removed 2 at a time because they're being expressed as homozygotes and taken out 2 at a time. That makes selection more effective and then reduces the load by a factor of 2. So that you get the half, right? Okay. Alrighty. Yeah. So these things I just said before. So what about inbreeding depression? We talked about that the other day. Just quickly show you a slide for this. We're back again to an honest graph that goes from 0 to 1. So you can read this as it looks. And you get substantial reductions in inbreeding depression for the alleles that have weak dominance coefficients, okay, regardless of whether it's hard or soft selection. And that's just because there's fewer of those alleles in the population. Selection was more effective at removing those weak alleles. And if you remember, this is a version of the graph I showed you last time. It is exactly those mutations of low H values that are the ones that are contributing most to the inbreeding depression. So this should have a fairly strong effect on inbreeding depression. All right. So we should expect load to be complicated, often reduced, but not necessarily with soft selection. And we'd expect inbreeding depression to be reduced under most circumstances. Okay. So that's kind of a natural break. You may have questions before we go to the next thing? One thing I'd like to mention about those previous slides, those are contrasting it to a completely panmictic population. In reality, what we might be comparing it to is not something that's completely panmictic, right? Most populations have some extent of inbreeding, or either at the within or among population level. A lot of this reduction in these allele frequencies may have already occurred, even in what you might think of as the outbred population to standardize, right? So it's not necessarily easy to look at empirically. Okay, so we've talked a lot. Many of us have talked about fixation probabilities this week. I don't think anybody's talked yet about how that's affected by the existence of population structures. So let me spend a few minutes on that if I can. Okay. Did somebody talk about it and I missed it? Nobody's willing to say. Good, we'll go on. Okay, so this is a little bit repeat. So approximately for Wright-Fisher model, holding or found this 2s. Kimura found a more general equation for it, but it approximates to 2s times effective size over the census size. And as we've seen, Kimura's more general equation, not this one, but the more general version for which this is a reduction, will let you deal with deleterious mutations. I think we've talked about that already. I think the first paper that tried to talk about what's the probability of fixation in a spatially structured population was this one by Murayama. And the title of the paper was An Invariant Property of Subdivided Populations. And what he found in that paper, considering models like the island model, was that the probability of fixation was 2s. And the implication of that was there's no change in the probability of fixation for when you have population structure. Okay, it would be 2s without with a panmictic population, it would be 2s in the island model. So there's no effect; it's invariant. Okay, and that's true for the island model. It's not. He was right about that for sure. You can get a little bit more general about that and ask, what's the probability of fixation with population structure? And if you write it in a slightly different, I'm going to write it in a way that it looks like the Kimura result, and it comes from the same logic as the Kimura result. The probability of fixation in a spatially structured population with soft selection is 1 minus FST times 2s NE over n. Okay, this is assuming actually that additive beneficial alleles for this simplified version of this. Before, we haven't needed to specify that it was additive. We just needed to use the fitness benefit of heterozygotes. That's no longer particularly true in structured populations because that the degree of structure determines the fitness of those heterozygotes in different ways. So you can write the equations for this and with arbitrary dominance, but this has a nice parallel to what we've seen before. I just want to leave you that. Okay, so this 1 minus FST term, you have NE over n. Now buried away in there is that the effective population size of the whole set of populations is affected by population structure. Okay, it's not just the same number that it would be if it had no population structure. So what's that NE? Well, that's something that Nick Barton and I did a while ago. Before I get there, so here's the NE for the island model. This comes from Wright and re-derived a couple of other times. This is the local population size times the number of populations. So this numerator here is just the total number of individuals divided by 1 minus FST. Okay, so that's the strict island model, right? It turns out, so your question yesterday about what's n, is n a fuzzy thing like biology, or is it just n? There's a real consequence here because the theory assumes it's n, and it's not stochastically n, but it's n. You have to say it with that emphasis, n, right? And I'll show you from the next slide why that matters. Well, but before I do, let's just say this: if you allow the contribution of each deme to just be predictable by the summing up the reproductive success of the individuals in the deme, and they don't affect each other, so like with a hard selection model, this is just ND. There's no 1 minus FST at the bottom, right? So this reduction, or sorry, this increase in NE for this island model comes from the fact that the populations as a whole are constrained to have 0 variance in reproductive success among populations. Every population contributes exactly equally. Now, if you think about the logic of the effective population size, effective population size is inversely proportional to the variance in reproductive success. So we've taken some of the potential variance in reproductive success among individuals and constrained it out of the system, right? So we're not allowing those individuals to all reproduce independently. We're constraining the sum of their reproduction to be n. That reduces the effective population size by some amount and leads you to an increase relative to panmixia of the effective population size. Okay. Yes? Yeah. Yes. Do you hear, Brian? Right. So the any, in this sense, has a couple of meanings. We actually derive the thing on the next slide as a asymptotic NE, as looking at the eigenvalue of allele frequency change over time. You get the same result by asking what's the coalescence time of two alleles taken from a metapopulation randomly. Okay. Yeah. Yeah, no, that's right. So Brian showed you that for diploid populations, NE was 4n over v plus 2. Okay, so that v was the variance among individuals. Yeah, so this is so this is assuming that each local population has an effective size equal to the census size of that population. So there's no variance among, there's no extra variance in, the variance among individuals there is 2 times 1 minus 1 over n, which is effectively a Poisson variance. Okay? So I think you can get something similar if you stuck in a local effective size here. You should get something close to the right answer. I don't know exactly the details of that. But this is given assuming that each local population is an ideal population. Okay? So what you could calculate its effective, the local population's effective size by that 4n over v plus 2. Okay? So this v that I'm talking about, which is 0 here, is not that variance among individuals within populations. It's saying there's low variance, 0 variance among populations and the total contribution of those. And so that would be a fraction of the among individual variance. That's reduced, that's eliminated. Okay? A more general version of this, and unfortunately just didn't follow this, this v means something different. So v in the equation that Brian showed you was the variance among individuals in reproductive success. The v in this equation is the variance among demes in their reproductive success. Okay? So it's the variance in the per capita success of demes on average, right? So some demes do really well. They're upstream and they're sending lots of migrants downstream. And some demes have very poor success because they have nowhere to send their offspring. That would contribute variance to reproductive success among demes. Or some places have lots of food and some places have little food, variance to reproductive success. Some places go extinct and others don't. High variance to reproductive success among demes. Okay? That's what this V is. And so there's a more complicated version of this that allows for other stuff to go on, but this is a easily digestible version. And you can see these V's are here. And if you plunk into this, the variance among populations that you would expect just by Poisson summing, the sum of the contributions of n Poisson individuals, you return to ND. Okay? So if there's no excess variance, if you're not constraining the variance in reproductive success among populations, you return to the census size of the metapopulation. If there are forces that increase over that Poisson expectation the amount of variance among populations of reproductive success, good places, bad places, extinction, no extinction, that's going to decrease the effective population size relative to the panmictic case. Okay. And I say it's still, I guess, 15 years after we first published this, it's still an open question what this variance in reproductive success among demes is. We don't know. Right? So we're certain it's not 0. Right? That makes no sense that it could be precisely 0 we know there's a lot of forces in many populations that create a lot of variance of reproductive success. So it's the simple island model conclusion is certainly not right in and of itself, but it may be actually in the wrong direction. Right? Question? Okay. Oh, right. So here's some. Here's what we'd expect in an extinction colonization model, the probability of fixation to be. Here's 2S that Maruyama said would be invariant. I'm being unfair to him because he was talking about a particular class of models, but it's not an invariant property of all population structure. And here's the prediction from the theory, and the red dots are results from simulations. It does okay. Okay. So this is a bunch of different parameter sets, all mixed together, different migration rates, different extinction rates, just plotted down as how well does it do overall. Works well. Okay. So it can deviate quite a bit from the island model expectation. Okay. So population subdivision, I think, typically reduces the probability of fixation of beneficial alleles. I didn't talk about it, but subdivision tends to increase the probability of fixation of deleterious alleles, and the reason for that is based on the thought that the effective population size is probably actually reduced rather than increased by population structure. Again, that's an open question, so this is actually worded too strongly. It's entirely possible for deleterious alleles to fix more rapidly in structured populations. But importantly, at a broader conceptual level, the fixation of these selected alleles is not invariant, right? It's something that depends on the details of the demography and history and reproductive success patterns of the populations that we're studying. So let's not do that. Wait. Okay. So we'll shift gears again. Anybody got any questions before we? Yeah. Yeah, that's right. Yeah, it does. So that's the fun part, is that all those details, to a large extent, just get reflected in what's the patterning of genetic differentiation among populations. Mm. Yeah, that's right. Yeah, so it's kind of like NE, right? So let me start from a different thing. I didn't do a good job last time. I told you I was a bit too negative and it wasn't very positive. Right? So I told you what things that didn't work, and I showed you all the island model, and all these deviations from the island model, and I drew you cartoons of those. And what I didn't do a good job of, it was saying that almost all those cartoons that I drew, we have equations that predict FST for them. And if we don't, we have methods to generate that FST. Certainly, we can do it by brute force recursion. And it's not that hard usually to figure out what it might be at equilibrium. Yeah. Yeah, and that, right. And the claim is that doesn't matter so much. It's the pattern that results is what, the pattern of covariance of individuals in the same population that's reflected by FST is the important thing for determining these forces. And so I like to, in this context, I like to think of FST kind of like NE as a sort of bottleneck parameter, right? Not a parameter about a bottleneck, but it's a bottleneck in the intellectual flow. There's a lot of different ways that you can deviate from ideal populations. There's a lot of different ways you can create population structure, but those can be described in one or two parameters, NE or FST, and then used for a broad variety of other questions, right? And to the extent that that's possible, that's amazingly useful. So instead of having a list of 47 different demographic things we have to think about, and a list of 112 different biological processes that we want to study, we don't necessarily have to study 47 times 112 different things. We can figure out these 47 things and how they make NE, and then plunk them into the 112 different things to say how does drift matter for evolution. Right? And I have to be careful that we're, check every so often that it's still working, but that's the power of this as an approximation. So I'm using FST in the similar sense. It collects the important bits of the information from the population structure. Okay. All right, so I'm going to briefly talk about something that I've worked on for a few years ago, but never published. But I just want to show you another context, a very important context, I think, in which population structure matters to evolution, and that is frequency dependence. So frequency dependence is when the fitness, the relative fitness of individuals depends on the individuals around it, right? Depends on the frequency of the other individuals around them, right? Now, I'm actually more interested in frequency dependence for things like butterfly coloration and mimicry, et cetera. But a very common context, common in the sense that people study it, is in terms of the evolution of cooperation. So cooperation tends to be a frequency-dependent trait. It's like your fitness as a function of how well you cooperate depends on who's around you. Okay. So I'll use that by way of example today. Right. So there's we talk about cooperation. There's a classic, beautiful result that has a lot of power, even if it doesn't do everything. That's Hamilton's rule. Hamilton's rule says that if you take the relatedness of individuals that are interacting times the benefit of a recipient of some act minus the cost to the donor, to the actor, if that's greater than 0, then that action will increase in frequency in the population. Okay? So basically, it's saying if you do something that might potentially help someone else, that in this case carries a fitness cost to you, and then you have 100% genetic similarity to yourself, so that's weighted by a 1 that you don't see up here for what it costs your genotype's fitness. But to extent R, the individual you're interacting with has the same allele that you do, right? And if so, the benefit to your genotype is increased by a factor proportional to the benefit, right? So that's so if this way to improve the effect, if this way to improve the fitness of genes like yours outweighs this effect of genes like yours, then it'll evolve in the population. That's Hamilton's rule. So you can rewrite this relatedness in terms of FST. This is not new, but useful just to connect the two dots. With haploids, R equals FST. I should say everything I talked about up to this point was, in fact, diploids. You can do them for haploids, but don't get confused. I've been diploid so far. Relatedness is just FST for haploids. It's 2 FST over 1 plus FST for diploids. Okay. Okay. So, but I'm going to stick in the FST world. I'm just saying there's a connection to that relatedness. There's a classic. Where are you? There. Yeah. We saw this the other day. The prisoner's dilemma is a so-called 2-player game. 2 individuals interact once in this case, and there's these R's and S's and T's are the fitness effects of a cooperator interacting with a cooperator or a defector interacting with a cooperator, et cetera. So these are what you get as a cooperator, what you get as a defector, depending on who you interact with. Okay? And prisoner's dilemma, by definition, is when T is greater than R is greater than P is greater than S. Right? It's always better to be a defector no matter who you're interacting with. But even though the highest fitness in a population would be if it were filled with cooperators. Right? So you have this conflict between the effects on mean fitness of individuals, or the effects of fitness on individuals and the mean fitness of the population. Right? It'd be great if everybody cooperated, the world would be one big happy place, everybody have a high fitness, but that's invadable by, in an unstructured population, that's invadable by a defector who would then go to frequency of 1, ruin it for everybody. Okay? Okay. There's other two-player games that you can also express with R, T, S, and P, but not necessarily with a condition at the bottom. There's what's called a snowdrift game. At least it's called that in Canada. I don't know what it's called the rest of the world. The snowdrift game is based on the notion that you're stuck in a car and a neighbor is also stuck in the car, and one of you has to shovel the snow to get out, but not both of you, right? So you're better doing it than not doing it, right? So you're better off doing it even if you're paired with somebody who's not going to do it. It doesn't. But if you're both, but you'd rather the other guy do it. Okay. So a cooperator gains a benefit, but not as big a benefit as a defector does when those two are interacting. Okay. And I'll show you some results about that game in a second. It's another kind of cooperation. Okay, so here are the conditions, and these aren't specific to any one of those games, but just to the different P's and S's, R's, and T's. These are the conditions for the evolution of cooperation when it's rare. FST has to be greater than a certain relationship between those. And here's the condition when it's common, for it to increase when it's common. Right? And it's possible, under some games, for it to increase when rare and not increase when it's common. Right? That would lead to a polymorphism that was stable in the population. Okay, so these are actually expressed for hard selection with the kind of equation. Oh, I should go back a step. The reason I got into this was dominance at the allele level. So dominance at the diploid individual level is not frequency dependent. Right? A heterozygote always has the same fitness relative to one of the homozygotes, regardless of frequency. But dominance at the level of an allele is frequency dependent. Right? The fitness of a big A allele relative to the small A allele depends on the frequency of big A's and small A's in the population. So the same style of equations that led me to the stuff I talked about a minute ago, with slightly more complications, can take you to these kind of equations. So yeah, so here's just one example. This is the snowdrift game. You can show that you can get cooperation, intermediate cooperation with, I think it's a snowdrift game. I'm really not 100% sure. If FST is large enough, right? You can evolve cooperation if FST is large enough. Okay. So the population structure matters. It matters whether you're paired with someone like you or not. And population structure increases the probability that you are paired with someone like you. Now, I don't have the slides to show you about this, but this is hard. I talked about hard selection. Cooperation is harder to evolve with soft selection for the same reasons that we talked about earlier. With soft selection, you're competing with relatives for access to the next generation. So if you're a cooperator and you do well, you're in a population where there's other cooperators, you do well, that leaves less resources for them or vice versa, and the response to selection is diminished as a result. And one really cool thing for the island model, and it's cool even though that's the island model, with island model and soft selection and where NE equals N, you get something called the Taylor model. Under the Taylor model, the effect of competition exactly counterbalances the benefit of cooperation. Right? So the benefit you get by interacting with other cooperators turns out under that set of assumptions is exactly counterbalanced by the competition between local individuals, and there's no net effect of selection on discriminating between cooperators and defectors. Okay? That goes away when you break any of those assumptions, but it still shows you how strong that effect can be. Right? That competition between relatives can actually completely eliminate what would otherwise look like a major effect, a major prediction. Yeah. Yeah, so these models are assuming it's local and pairwise. Right, yes, they're assuming local and pairwise. They aren't necessarily assuming that you only have a single pair per lifetime. Right, so you could be going about your business and continually pairing up and making interactions with other individuals. But they are assuming that all of those subsequent interactions are independent of each other. So there's no reputation, there's no, right? now, the general mechanics of this are kind of agnostic to that. If you can write a frequency-dependent equation, if you can write an equation for what's the fitness under a particular game, however you want to play it, multiplayer game, repeated interactions, reputation, indirect, blah, blah, blah. If you can write an equation that says, here's the fitness in this circumstance as a function of allele frequency, you can plug it in here. Now, so far, this only gets up to a certain order of polynomial of that fitness function. But in principle, you can do it. Yeah. So what's missing in the Hamilton's rule equation isn't really missing. You just can't see it. It's that with Hamilton's rule, you really need to consider the all individuals who are affected by a particular interaction, right? So, and what you can see in the classic textbook version of it, you've got the guy doing the action, you've got the direct recipient of that action. But what you also need to consider is anybody else who's affected by that interaction, in this case, by that indirect competition, and their relatedness to the original actor. Okay? So really, there's other terms. There's one other term for every other individual in the population, and they all get a minus b over n deficit caused by that competition term, or minus 1 over n, b over n deficit, times the relatedness of them to the original, and that winds up adding up to exactly counterbalance the benefit to the recipient. Right? No, because it's well, what it depends on, it turns out, and Dave Van Dyke and I wrote a paper about this. In the Taylor derivation, he conflates the effect of n for divide. When you assume island model, there's the same n, but they actually have different roots. So there's an n that talks about dividing up the effect of the competition, and that's a census size. And there's an ne, which is proportional to the relatedness. And when those aren't the same, you can still create the avenue for evolutionary cooperation, even under the same circumstances. Yeah. But okay. So with spatial structure, kinds of cooperation become possible that weren't possible in panmictic populations. You can get coexistence of cooperators and cheaters, which is difficult sometimes without that structure. And crucially, it depends on how individuals, where individuals are in this hard and soft selection continuum. Right? The closer to hard selection, the easier it is to evolve cooperation, okay? All else being equal. Because you're not competing against your brothers and sisters who are also cooperating. Okay. All righty. Questions? How are you doing? Okay. So I want to finish, I think, in the next 20 minutes or so, on a completely different topic. We're still talking about population structure, still talking about spatial stuff, but really quite different from what we were talking about before. We'll now get more statistical genetics-wise. How do we find genes that are responsible for local adaptation? So I told you last time about reciprocal transplant experiments. That's one way to identify that there is local adaptation. Right? You can potentially do that also by more indirect ways of asking whether traits which we know something about the mechanistic function of are correlated with environmental axes, et cetera. We start addressing local adaptation. But how do we know which genes may be responsible for local adaptation? Okay. Now, and there's kind of, I think, two high-level approaches to this. One is to do something like a QTL experiment where you have lines that are adapted, for one reason or another, are adapted to different environmental circumstances, and then you do a classic quantitative QTL experiment to find the genes that are causing the differences between those populations. Okay, that requires that you know what traits are adaptive, but there's a lot of times when, of course, we know that. Okay. But I'm going to go at a earlier level than that in terms of the kinds of study that you could do, kind of closer to the real organisms, closer to the populations in nature, this is all inferential. It's all non-experimental, right? So this is all observational studies, correlational studies. And a lot of the work I'm going to tell you about was done with Katie Lauderhouse, who's now at Winston-Salem University. She was in my lab before. So now let's look at, we're going to think about these correlational approaches, approaches that try to find what genes in the genome are likely to be experiencing spatially heterogeneous selection. Okay? We're going to, all the approaches that I'll talk about from now on do not require that we know what trait is responsible for that local adaptation. Okay? So we're gonna know where things are. We might know what it's like where they are, and we're going to have genetic or genomic information about individuals that came from those places. Right? We're not going to look at the phenotypes in between. Right? So this is about what genes relative to spatial structure. So within that realm, there's kind of two broad approaches to look for. Sorry, these are two, but they're not the two. Oh, there. Good. Sometimes they make more sense than I think. So there's two broad approaches for identifying what genes might be responsible for local adaptation. Okay, now, I'll talk about both briefly. One is you can lump under the label FST outlier approaches. Okay. FST outlier approaches say, let's look at the FST of lots of loci. The ones that are most likely to be under spatially heterogeneous selection are going to be out here in the far right tail. Okay? Because those are the genes which aren't just experiencing genetic drift, they're also potentially experiencing, well, so if a gene was experiencing drift and was experiencing a spatially heterogeneous selection, that would drive the allele frequencies to be different between populations that would give you a high FST. Okay? There's no guarantee they'd be out there, it's just where we want to look for them. Okay. So that's the FST outlier approach. I'll talk about more of it in just a second. The other approach is coming on gangbusters now. People are doing lots and lots of cool things with this. Doesn't really have this name. It's just a name we used for a while, but it'll suffice. It's looking for correlations between the environment where genes come, where individuals come from, and what alleles they have. Okay? So in a perfect world, where you didn't have to worry about lots of statistical details, you could just think, oh, let's go out and I want to find a gene that's a high frequency when it's wet and low frequency when it's dry. So I could just look at a correlation between gene frequency and the environment. And if you were able to correctly make a lot of assumptions, that would be all you'd need to do, is just look at a correlation between the environment and the allele frequency. That's not what you can do, though, because in these cases, that correlation suffers from the fact that there's spatial autocorrelation of both the environment and spatial autocorrelation of the gene frequency. Okay? And so when you have, that creates a non-independence for your data points, your samples, non-independence for your x and y variables, and you can't do just simple regression. Okay? Because you're going to find a correlation sometimes in that case, just because sometimes the allele frequency goes from high to low from east and west, and sometimes it gets rainy in the west and dry in the east, and that'll give you a correlation, but it's completely not necessarily caused by the selection. Right? Just by chance, you're going to get that pattern quite a bit. Okay. So two broad approaches. They're both looking for alleles responsible for local adaptation. One of them is just saying, are these alleles, are these loci unusual in how differentiated they are? And the other is potentially more powerful, and turns out to be definitely more powerful, is saying, well, we're not just asking, are they more different for this locus, but are they more different in a patterned way that you might predict based on the environmental data? Right? So that potentially has a lot more information. This one, you can be agnostic about what the environmental variables are that matter. And that's a really powerful thing, because we might think we know, but we don't. This one, you have to know what is likely to be the environmental axis that's important. Okay? So let's explore a couple of these. I'm going to show you, we're going to, in passing here, show you some tests of these methods. And I'll talk about the specific methods more in a second, but I'm going to show you some tests of these methods based on computer simulations of large landscapes of digital organisms. So we're going to test the island model, even though we don't believe in it. That's one of the things that's assumed by some of the methods. So we'll give it there just as a baseline. We have an isolation by distance model. So this is a case where the individuals move according to a Gaussian distribution centered on where they were born. Okay? So in the island model, if an individual moves from here, it can go over here just as easily as to the next site. With isolation by distance, movement is fairly local, right? And it has a tail going out. But almost all individuals stay nearby. It's almost impossible to go from here to here, except over many, many generations. Right? So we expect a lot more genetic patterning here. And then we also did 2 non-equilibrium cases where we expanded the populations from a refuge. Okay? So we come from a place where there was glaciation until 10,000 years ago. All the species around us weren't there. It was buried by almost a mile of ice. And so everything that we see there moved back there from some refugia further south. Okay? So this is part of a larger project on lodgepole pine. And so this was of particular interest to us, both for the species and geographically for us. So we did these non-equilibrium models of expansion from either a single refuge or 2 refugia. Right? So these are coming back into secondary contact later and provides an opportunity for a patterning over the landscape. Okay. Now, we chose the parameters of these models so as to make FST identical or nearly identical for these different scenarios. And the reason we did that was that FST is easy to measure. Right? So if you're an empiricist applying one of these methods, that's not in doubt. You know what FST is. It's very easy to measure. So what you don't know is what's the demographic history. Okay. So we're going to condition, effectively, we're going to condition on a constant FST. Right? Okay, we know that, 0.05. But we don't know how do we get there. Okay, so that's the logic there. These are the distributions of FST for these different models. I should say these are all neutral loci. Right? Later we put in selected loci, but what I'm showing you here, these are all neutral loci. Okay? Now, I told you the other day, yesterday, that's another day, I told you yesterday that the FST should be the same for all neutral loci. Right? As long as they all, autosomal, et cetera. I didn't quite say that. I said the expectation for FST would be the same for all loci. Okay? As it turns out, FST can be quite heterogeneous from one locus to the next. And the reason for that, let me just draw real quick. The reason for that is both statistical, right? So if you take a sample and your FST is the sample, then there's noise in that estimation, and so it's going to look different from one locus to the next, and that's going to be true in real data. But on top of which, if you have a finite number of populations, and I'm just going to draw, for example, here's tracing the allele frequency in one population, and here's the allele frequency in another population. I'll just stop with there, but you might have more and more and more of these. These are just drifting over time, right? It's a constrained drift, perhaps, because they have dispersal, but they're not. The allele frequencies are changing over time in any given population. There's going to be some times where they, by chance, have drifted to a similar allele frequency, and FST will be low at that locus. And there's going to be other times when they drift to different allele frequencies at that point in time, and they'll have a higher FST. Okay? So if you have a finite number of populations, this effect creates variation, real variation, in the FST of your populations. It gets smaller and smaller and smaller as you have more and more populations that you're studying. Right? If you have a lot of populations in nature but only a few that you're studying, you have this problem, but it becomes more of a statistical than a biological issue. Right? You've sampled into populations that have a higher FST, but it amounts to the same thing in terms of the difficulty, right? So we're going to see variation in FST even for things that are evolving the same way, right? It's a stochastic thing by its nature. We're going to see variation. So here's what it looks like for an island model. It's hard to tell from this, but it's going to, it fits very well a chi-square distribution, FST over the mean FST times the number of populations minus 1, has a very close to a chi-square distribution for the island model. But here's these other cases. You can see this one is a little bit broader. The tail is a little bit thicker. You have to squint a bit to see it there. But here's the refugium models. You can perhaps certainly see that it's broader and the tail is thicker. We've exaggerated the tails here. And you can see how many more loci are out in the tail with the refuge models than they are in the island model. Okay, so right there, we're going to have a problem. A lot of these methods assume that you have an island model, and to one extent or another, when they do their calculations. And so we're going to expect to see a lot more false positives for these non-island model demographies because the shape of the distribution doesn't match what we would have thought. Essentially, this is because there's effectively fewer populations in the evolutionary history of these refuge models. Right? So there's fewer populations, it becomes a broader distribution. Right. Okay. I won't belabor this difference. There were, when I made this slide, a couple of important FST outlier tests. There's another one which I should be showing you that does quite better than these two. It's called FLK. FLK works quite well. But these two, the classical outlier test are FDIST, which basically takes the distribution and simulates an island model based on the FST of that distribution, and then asks where's your locus relative to that simulated set of data. And Bayescan, which is a Bayesian approach based on Dirichlet, but it's basically assuming that all the populations go back to a common ancestor with no migration between them since that. Right? So again, doesn't match the biology well and doesn't have any method to account for the complicated dispersal patterns that we might see with isolation by distance. And let's cut to the chase here. What we see for both of these methods, these are the number of outliers out of 10,000 neutral loci. Right? These are the ones that are flagged by the method as being under selection. But we know how many were under selection. In this case, it was 0. Okay? Because we did simulations, we know the answer. It should be 0 for the island model. Right? This number ought to be around 0 to 1 because of the way that the correction for the false discovery rate corrections are done. But Bayescan is not even perfect for the island model. But look over here. We're getting as much as a quarter of the genes that are being flagged as under selection under this 2 refugia case because the real population demography doesn't match what's assumed by the method. Okay. Now, part of the reason for this pattern is something called allelic surfing. It's not actually a huge part of it, but this allelic surfing is so cool. I want to spend a minute telling you what it is. So let's take a little break. This is something that I guess was originally mentioned by Luca Cavalli-Sforza, but it really got made into a real thing by Laurent Escoffier in Geneva. It's just one of the coolest things, I think, ideas that's come up in spatial population genetics recently. So the idea is that you start from some founding population, and then that expands over space, right? So at the front of at the front of that wave of expansion, there's some distribution of population size. Okay. Now, if you think about it, assume that there's some kind of local density regulation. So that means the individuals out here on the tail, the ones right here at the front of the wave of advance, are super important ones, right? And they're super lucky. Right? They're in a place, it's good habitat, but hardly anybody else is there. They're going to have a ridiculous reproductive success. Right? They're going to have lots of offspring on average. Not only that, where does the next wave of advance come from? It comes from the front to a large extent, right? So the guys here who have a huge reproductive success not only contribute a lot to the next generation, but they're the ones who contribute to the subsequent population spatially later in the wave of advance, more than everybody else. Okay, so what happens if right there in that individual or in a recent ancestor there was a new mutation? Well, that new mutation is going to increase on average in frequency a lot more than if it happened back here. It's basically going to hitchhike, not necessarily with another gene, but it's going to hitchhike along with the kind of just the ecological reproductive success of those individuals. Okay. So now Laurent's talked a lot about this in terms of the fact that then you can get allele, kind of a sudden change in allele frequency. That doesn't happen all the time. It's a rare event. You don't expect it to happen all over the genome, but it happens enough to really confound our understanding of spatial structure. So one of the ways that you can get these false positives, you get this neutral mutation, rises to high frequency, and it just has this pattern that looks just like local adaptation, right? So you come along to here, suddenly you have a high frequency of an allele that wasn't there before. If you look at it at the molecular level, it shows all the signatures of selection because it's increased rapidly in numbers, so it hasn't had a lot of chance for recombination with other types. So it'll be give you a haplotype block of low variation. Right? It'll look like selection, right? But it's not. It's just it got lucky to be in the right place at the right time, right? So that's one of the ways that we're going to get false positives in these expansion models. Okay. Then we also plunk these populations down onto different environments. Here's just some pretty pictures of what those environments were like, and use that for loci that did have a selective effect on those environments. And we found that actually the methods have pretty good power to detect alleles that actually contributed to the local adaptation. Right? Not all of the loci that could contribute did, but if they did contribute to local adaptation, there's a pretty high power for detecting them. Right? So that's good news. The bad news is if you looked at the mix of alleles of loci that were flagged as being under selection, the false positive rate is actually 50 to 70%. So half to 3/4 of the genes that were flagged as being under selection were actually false positive. So that's, depending on your reasons for doing the study, that may be perfectly fine, or it might be unacceptably high. Right? You can have a lot of false paths to go down as you started trying to trace these down to see what to do with them next. Okay. And I'm running short on time. So this is results for, I think, one of the most effective of the environment allele associations. It's a program called BAY&V. This is by Graham Coop and his collaborators. Apparently it was named BAY&V because he lived in the Bay Area and everybody was envious of him. So that was that's what he told me it was named from. If you go to France, people refer to this as Bayonf, and you have no idea what they're talking about. But so BAY&V apparently is the right way to pronounce it. So this is a way that looks for the correlations between gene frequencies and the environment, but corrects for the pattern of association caused by the autocorrelation of allele frequencies, right? And it's not fixed on autocorrelation. It uses the actual matrix of genetic relatedness between pairs of populations to correct for the regression that it's doing. Okay, so BAY&V does a good job of detecting the alleles that matter, these ones with stronger selection coefficients, and has a perfectly acceptable false positive rate. Okay. I'm just going to say this without telling you much about it. We've actually developed a new method for FST outlier approaches, and the short version of it is that it works. and you get appropriate false positive rates. The slightly longer version of it is what we noticed was that these distributions of FST weren't the same as you predicted from an island model. These other demographic histories gave you distributions that weren't like the island model, but they still had something that looked like a chi-square distribution. And so what we do actually is infer, we assume that it's chi-square, and we infer what's the parameter of that chi-square via likelihood. Okay? And to do that, what we basically do is we trim off the data in the zones that are most likely to be affected by selection. We take off the highest FSTs, we take off the lowest FSTs, and we use likelihood to say what's the parameter that predicts a chi-square distribution that would go with, knowing that it was truncated at those 2 points. Okay? So we can infer the right parameter from the bulk of the data, and then use that distribution as our null distribution. And it's completely ad hoc. I mean, it's completely, well, it's not completely. We have some logic behind most of it, but it's very inductive, right? We notice this pattern about the shape of the distributions, regardless of what pattern we tried to investigate, and we did far more than I showed you here today. It looks chi-square, and the proof in the pudding is when we apply this to simulated data under every circumstance we've applied so far, our false positive rates are dead on what they ought to be. Right? So we can account for these variations in population structure without knowing what that demographic history was that generated it by using the mean FST and using this estimate of the degrees of freedom of the underlying neutral distribution. So that's something we're writing up now. Yeah. Yeah. So right. So unlike the ones I showed you earlier, they don't need that many loci. Yeah, this one you need, kind of genomic next generation level replication. I'd say 1,000 or more loci. So it's something that's just on the cusp of affordability, depending on, I mean, on the cusp temporally. Like this is something that's going to be more and more easy for even me to do. I have a tiny budget, but I can even conceive of getting 1,000 loci now, and 2 years ago I couldn't even think about it. And I think that'll keep moving along. Okay, so let me wrap up. We got 2 minutes, and I just want to return to a theme from yesterday, which is when we think about, especially when we think about applying models to data via the intervening step of somebody's statistical genetics machinery, be careful about what assumptions go in to those approaches, right? So if you're an empiricist, read the fine print. Read what does it assume, how well does that match, look for resources that tell you whether or not that matters or not, and really remember the biology, right? So if you're doing something with a principal component analysis, for example, that's taking you a couple of steps away from the actual biology because the axes don't necessarily have any intuitive meaning. Well, look at it again on the non-rotated axes to make sure that it makes some sense to you. Look at it trait by trait level. Don't just look at a structure plot. Look at it, the pattern of pairwise FSTs among populations, and try to envision what's really going on at a level that's more intuitive, or whatever it is intuitive to you. Try to remember the biology when you're looking at the results. Challenge the results when they make assumptions that don't match your biology. And if you're theorists, I say increase the font size on the fine print. There's a tendency when we're doing theory and we're doing statistical genetics to state the assumptions perhaps, but we don't overemphasize them. And then when we get to the second paper about a method, we just refer back to the first paper. And when we get to the third paper, we just assume everybody knows the world works just like an island model. Why would you ever talk about anything else? We got to really maintain focus on the difficulties and the challenges and the limitations of the methods that we're doing. I mean, I think every paper, regardless of whether it's empirical, theoretical, we should require each other to have a section in our discussion of what's wrong with what I just did. Right? What's the limitations? How could it be improved? What assumptions that I make that might matter? You think, oh, that'll keep the referees will never let me publish this. But in fact, at good journals, the referees, you'll just take the thunder from the referees. They'll say, oh, well, of course they assume this, but, they talked about that. I can't really slam them for it because they already said it, right? It's just better science, more open, more honest, and more communicative to your reader about what you really do know from what you did. And learn the biology as well. So it's fantastic. It's really fantastic when people come in from outside a biological background, and it's really wonderful, and that can be so much more valuable as everybody in this room, I think, is doing. It's so much more valuable when you learn the biology and not just come in cold. So thank you. I'll give you that one. Lunch? Questions? Lunch. That's me. No.